The third speaker in the session on Betacoronaviruses is Louise Cosby, who's come all the way here from Northern Ireland. And Louise is going to talk to us about calf coronaviruses.
Right, well, it's not totally on coronaviruses. It will have some feature of coronaviruses, but the disease that I'm actually, let's see, where's the forward here? Oh, right, I've got it now, thanks. So the disease we're of interest to us is bovine respiratory disease, or complex, as it's referred to also, BRDC. And this is really, it's one of the most economically important diseases for the agri-food industry, and it's the main cause of cattle mortality from 1 month to about 2 years of age, and costs billions of pounds globally. Really. So the initial insult is usually a viral infection and a number of viruses, and not all, and there may be several at once or one that starts. This is a respiratory syncytial virus, bovine herpes, bovine parainfluenza 3, and bovine coronavirus, which is actually becoming more prevalent at the minute, and bovine viral diarrhea virus, adeno, and few others, and then this predisposes the calves to secondary bacterial infections and mycoplasma, etc., and maybe some other viruses coming in due to the immunosuppression of the initial virus infection. So really what we're looking at, we do a lot of experimental infections in cattle, and we've done quite a few with bovine RSV and bovine herpes virus, and looking at genetic markers of susceptibility, etc. And a lot of that work is published with collaborators in Missouri and at Chagas and the USDA. And we are currently doing a big study with RSV to look at microbiomes in the respiratory tract and the gut in challenged animals. But today I'm really talking about natural infection, and we're interested in this. There's really not a lot out there in the literature to understand the response in the cattle to viral infections. They're also, what's the differences to the different viruses, and maternal antibody. Most dams are vaccinated, and then the calves get maternal antibody from the dams, and that's meant to give some protection, but there's very little known about that. And also to determine maybe which virus actually caused a pneumonia, a BRD, if we don't actually know, and to look at the clinical parameters around this. And this all informs, obviously, vaccination and treatments, etc. So we had the opportunity, I'll just go through this briefly, to look at 40 male Holstein calves in October 2021. These were unvaccinated, their dams would have been vaccinated. They were brought onto us for another study, and we usually select for study animals from these, but we couldn't actually use them for the study intended, because about half of them got pneumonia, 21 got pneumonia. So we decided actually with a PhD student, and could we use these for something useful? So we decided to actually look at the immunology in the actual natural infection. So animals were we actually couldn't swab them initially because they weren't under license, but we brought them under license in January 2022, and then we were actually able to take blood and that time point, and then we bled them again in May, and both time points with IgM, IgG, VNTs done, and also then in May we were able to, 4 months later, to do nasal swabs and to look at mucosal IgA. We put sponges up the calves' noses. So there's a few histograms here, but I'm just going to point out the main features, really. So if we look at RSV, first of all, you'll see these stars aren't significant. These are the calves which actually didn't show any clinical signs. So they may well be asymptomatic, but they weren't showing any clinical signs, and they didn't have to be given antibiotics. So just to see if they align with any of the immune parameters. So if you look at the IgM and IgG, which is the you'll see that about 30% and 70% of the animals respectively were positive for IgM and IgG, and this declined obviously a bit by the 4 or 5 month period, and but it didn't, you know, there were some animals still reasonably positive. When we looked at neutralizing antibody, there were actually 2 animals which were only had neutralizing antibody in January, and this went away by May. So most of this antibody was not neutralizing. If bovine herpes virus, a reasonably similar story, very little neutralizing antibody, and again gone by 4 months, and again decline of IgM and IgG. For coronavirus, a similar picture, except we seem to see an even more rapid decline of IgM and IgG in these animals, and no neutralizing antibody at all was found even in January. So seems to be less carryover of this probably in the maternal antibody, and that comes from the colostrum, which the calves are fed. So a bit of a difference there with the coronavirus. And then for parainfluenza 3, so what we think here is, because we've still got very high levels of both IgM and IgG, and IgM in cattle seems to last longer than IgM in humans. So that's quite an interesting point. Others have also found that. But what you see is the levels remain quite high. We also have quite a lot of neutralizing antibody, and in May, a lot of that is actually retained. So we actually think that the initial pneumonia or BRD that the animals got was probably due to the parainfluenza virus 3 from those kind of results. So it may be a signature. The neutralizing antibody is quite a marker of actual active infection. We've looked at mucosal IgA in May, and the thing to point out here is, okay, there are as you'd expect, maybe the PIV3 is still quite positive in May, but if you notice the bovine coronavirus, that is actually quite high. You remember the IgG and the IgM was low, so this is quite surprising that they've got quite high mucosal IgA, and it is known that the mucosal IgA, it can be coming from the maternal, from the colostrum, and it does get into the mucosa. So this is probably a passive transfer, but interesting, and obviously needs more investigation. We looked at gamma interferon levels, and what you actually see here, these are different graphs, but you see they look very similar, and that is because we were finding animals, when we look, this is done by ELISpot on the PBMCs, but we're finding that some animals are actually high reactors to all the antigens, not just a particular virus, and others are low reactors. And our colleagues in the TB field also suspect this when they're doing gamma interferon tests. So, but they weren't really sure about it. So this kind of shows that maybe you are seeing this overall reaction. Some of these animals just react to everything in a much higher way. For bovine, for interleukin 2, then we also looked at what you can see. All the animals have some of this, and this is actually, again, higher for bovine PIV3. So again, and it stays high. So it's probably, again, maybe relating to that being the actual active infection. So just to summarise then again what I've told you, so the majority of calves have IgG and IgM to each virus, but the decay is actually more rapid for bovine coronavirus, and there was little decay for bovine PIV3, likely because it was the active infection, though we can't totally confirm that. And there was mainly only neutralising antibody, and particularly in May, to bovine PIV3, and high responders, producers of gamma interferon. But the interesting thing which does need followed up is this mucosal IgA for the bovine coronavirus. I don't know what the parameter in humans, if that's been looked at for SARS-2, but it might be certainly of interest, and there are parallels. And the little dots, stars on all the diagrams I showed, there was really no correlation with any of the immune parameters in any one parameter. So what's stopping an animal actually getting overt clinical signs, etc.? We don't have an answer to that. So just concluding, PIV3 was most likely the circulating infection in the calves, due to the factors there that I've already mentioned. There are different antibody profiles in individual calves against each virus. That's really not been looked at before in individual animals. And except for PIV3, the majority of the animals to the other virus are likely maternal, and they're non-neutralizing and therefore unlikely to be protective. So there is probably limited protection, and it's known it doesn't give really good protection, just the colostrum, the calves not being vaccinated themselves. And the bovine coronavirus IgM and IgA levels, those differences to the other viruses, say, need further investigation as to why that is. We need to look at the antigens involved and etc. So the study overall has really given us further understanding of the markers of active and passive immunity and virus differences, but it's also raised a lot of other further questions. Thank you for your attention.
Thank you very much, Louise, in particular for sticking so well to time. Speakers in the flash talks note the good example you've been set by your chair.
Yeah, I had to make sure I did that.
Thank you. Right, we have one question at the front there. There's a microphone behind you.
Hello, so fantastic. Oh, Jesus. Fantastic talk. I have actually a question about the the bovine coronavirus infection in general. What's the outcome from a physiological point of view? Is it worse than the other viruses? Is it more asymptomatic, and that might also explain?
Not really, but with bovine coronavirus, which you don't get with the other ones, is the diarrhea. So, and there's people debate still whether there's strains which give you more respiratory infection and ones which give you more diarrhea, but most of the calves do get a bit of diarrhea, even if it's quite respiratory. And obviously the other virus, which I didn't mention, was bovine viral diarrhea virus, but we tested for that and none of them had antibodies to that. So there is an eradication program, so yeah.
Okay, thank you.
Yeah, down further back on the other side. Sorry. Dallin could have moved, but he's just stuck. But he didn't.
Thanks, Louise. I think you said that the majority of the antibodies here are likely, the calves themselves are not vaccinated, but the mothers were.
Yeah.
And I just wonder the nature of the vaccines. I think most bovine coronavirus vaccines are live attenuated, is that right? And I wonder if it's the live attenuation of that vaccine which might lead to the high maternal IgA transfer there in the colostrum.
Yeah, it's possibly, Darren, yeah. I mean, Dallin, we need to actually go back and look at that and actually get the vaccine records off the farmers, which we're trying to do at the minute. So, yeah, it might be something to do with yeah, as you say. But I think it's interesting because if we can figure that out, then it might be a way of giving better protection. The mucosal IgA, yeah.
Thanks. So, Julian, did you want to ask a question, or were you just running around to attract attention?
No, I was greasing Mars and I do have a question.
Right, thanks.
Louise, is there a lot known about either the sequence of bovine corona or bovine RSV? Because just thinking, David, behind me, for RSV, obviously you've got subgroup A and subgroup B in humans, and as immunity waxes and wanes, I guess one takes over from the other. So do you see that in cattle, and what's the situation with, I guess, sequence diversity of bovine corona, if anyone knows?
You asked me about corona, not RSV. So, yeah, I'm not sure. I mean, the bovine coronavirus, it is actually the precursor of OC43 that jumped into humans. Yeah, I mean, I'm not looking at it, but there are lots of colleagues are looking at the sequence of bovine coronavirus, and there are quite a lot of changes occurring as well. So it is mutating, and I think colleagues in the States that it has been coming up as a cause of BRD, much more so, and some of the more severity linked with mutations again. So there are quite a lot of parallels with SARS-2. I'm not sure that's what you asked me, but that's what
I think we should wrap up, so we'll stay on time there. I'm going to hand back to Julia, and thank you very much, Louise, for a great talk.
Oh, wow.